Okay, so here's the wheat physiology team, and some of these guys will be here. Francisco's the man who's running the drones. We'll get into that. Shivers, pre-breeding and molecular breeding. Hammer, photosynthesis, partitioning. Boss doesn't ever do anything, right? We all know that. Pancho, we call him. He's liaising with breeders, and that includes our own breeders, as well as the ones in Mexico, because we have quite a large portfolio from the Mexican government, some of which is supporting the bread wheat team. So he's there liaising with that. Carolina, automation. These are both actually Mexicans who came through a PhD program sponsored by the Mexican government. And Carolina St. Pierre, who will be here again later. She's coordinating some transgenic work and also the international phenotyping hubs that are, for example, one of them is here in Morocco for Drought. And Jacinta, so we have this project, the International Wheat Yield Partnership. So Jacinta, she's doing a lot there with logistics, and with a lot of visitors, and believe me, there's a lot of logistics. Okay, so that's the team. Which one was it? Leo? Oh yeah, there we go. So Lee and I came up with this program. What traits should we measure in the field? I mean, that's a really open end. We could talk for a whole day just about that, actually. But let's see, we'll have a bit of a go at it. High throughput and precision phenotyping. The world and scientists have a love affair with high throughput, but it's a lot more to phenotyping than high throughput. So I want to capture some of those ideas, and that's very much related to the technology. We all love gadgets and toys because we're scientists and children at heart, right? So again, there are important things to say about that. For example, high throughput. Drones, seems really easy to fly a drone, but it's not easy to process an image, and these are important considerations. And then a case study, in the case of our own program at CIMMYT, where we use pre-breeding, we're involved in pre-breeding, so that's what I want to talk about. So let's start off with which traits should we measure in the field. So a lot of that question is based on a number of other factors, like, for example, what's your target environment, obviously. And here you might have seen this already. These are the various environments of wheat in the world. So divided between spring, facultative and winter. Don't need to give a class on that to this group, I'm sure. And the fact that we're looking at different types of environments, high yield, heat stress, drought stress, high rainfall, which is more relevant to pathology, obviously, and so on. High latitude. So obviously that's the first consideration when you're deciding which traits, where do they fit, where's it going to go. I'm just going to press here, I think. And then it's easy to forget. And it's funny, really. It's easy to forget that we're not breeding for now. We're breeding for however long it takes to breed for, which is anything from 10 to 30 years, depending on the state of the art of the technology, right? And so this is a modeling exercise here, one modeling exercise, which shows the growing season, the average growing season temperature for a set of sites around the world. And what you see is that the red temperatures, the temperatures in red in brackets, the red dots are places that are already heat stressed. But the red numbers are the places that will be heat stressed in 2050. And there's a rather worryingly large number of those. So when we talk about traits, let's remember that we also have to think about the future, not just our breeding stations, which of course is all too easy to do. And here's another modeling study. This was by the AGMIC group, and they took a set of sites and they put +2 degrees centigrade into the model and calculated that basically models proposing that or suggesting that 6-degree decline in productivity per degree of average temperature, which is again a worrying figure when you look at the predicted climates in the future. And that doesn't even take into consideration heat shock. Because a heat shock, you can have the same average temperature. With a heat shock, you can also lose a lot more than 6% here. So coming back to our stations, then we have to think about which traits can we measure based on the temperature profiles we have. So this is Oregon, which we always say is the mecca of wheat. Maybe not everyone agrees with that, but we could have that debate in the bar, I suppose. And so you have different sowing dates for different types of environments. And yeah, you can simulate, for example, if you sow very late here, you can almost have an increase in night temperature with not too big an increase in day temperature. You could select for night temperature, for example, and you're sowing here is selecting for warmer conditions. So very much the traits that we can phenotype is a function of temperature. And then I mentioned heat shocks. Well, there's no field environment in the world where you can reliably get a heat shock, especially not at an exact development stage where you think it may be important. So we have to modify environments. So these are some heat tents that we've been playing around with, and other groups do it the same way. You can establish a small 2 to 3 degree increase in temperature by playing around with the ventilation in one of these tents. Is that high throughput? I don't think so, but it doesn't mean it's not important. So these are aspects of the environment, some of the traits. Let's talk about some of the traits. I don't think we'll spend a whole lot of time on this because there are many traits and everyone has their favorite trait. And like I say, they are going to depend on the environment. But here's some of the traits that we actually do select for heat, the warmer environments. Actually, when one of the effects of temperature is to, as we all know, is to accelerate growth. And so because of that acceleration, I don't think it's really understood perfectly well why. But you end up with smaller leaves, smaller organs, shorter plants. And presumably that's because as things get accelerated, there's not really enough time to pick up all of the nutrients and so on. I don't think it's particularly well understood. So one of the big effects of heat, especially if it comes early, is that you just never really get enough light interception. And so you're always kind of behind on the curve. And you never catch up. Chlorophyll density, the vascular system is important. We've shown many years ago, decades ago, that cooler plants do better not only under drought but also under high temperature conditions. And this is related to the ability to match evaporative demand in a hot, dry environment. Spike photosynthesis, I'll come back to that one, that's a nice one for heat. So light interception, as I mentioned, some photoprotection when you're at high temperature, just as it is for us. At high temperature you get sunburn, and plants kind of get sunburn, and leaf wax is sun cream for plants. And pigments is a way of potentially transmitting more energy, so you don't burn. That's depicting a lighter shade of leaf, pale green. I think there's land races, maybe even from Morocco, that show that characteristic. And then of course, these are traits related to light interception and radiation use efficiency, and then there's the partitioning, the all-important partitioning. Very often under stress, plants don't express sufficient harvest index. So these are some of the traits that we consider for making sure that the biomass gets into the grain. Stem carbohydrate storage in the stem mostly, and remobilization, grain set and sterility, grain size. All of these traits. So like I say, these are the traits that we actually use in pre-breeding and to some extent in breeding, because you see genetic variation for these. And we can go to, now I'm moving around again, and we can go to a more complex model, so more theoretical. Some of those same traits are here. but things related, for example, to rubisco that are very much still in the experimental stage. Rubisco is what photosynthesis people call a promiscuous molecule, because it will go with and fix CO2, but it will also go with oxygen. And the warmer the temperature, the more it will go in the favor of oxygen, which is not fixing carbon. And so there's research going on to understand better and measure the genetic diversity for specificity, that is, for CO2. But these are not things that we can put into breeding yet, because they're still in the experimental stage. Respiration, there's a big one. We all have this notion that respiration, especially the warmer the temperature, especially the warmer the night temperature, is a problem, but we don't really know that, actually. It's a speculation. There's a little bit of evidence, but not very convincing. Many areas, for the younger people here, there are so many things that we don't know which traits we should use. There are exciting research opportunities, in fact. So some of the same ones are here, the light interception and so on. Signaling, that's an important area. Plants, typically, well, obviously crop plants, like any other plants, have been evolving for many millions of years. And in order to survive, you have to be conservative. Because if you're taking risks all the time, sooner or later you end up as a genetic dead end, an evolutionary dead end. But actually crop plants, we don't want them taking risks. We don't want them to be so conservative. We don't want them signaling, sending out signals saying, oh, slow down, let's not take the risk. Because we take care of them. We irrigate them, we apply fungicide, etc. And so this issue of signaling, I think, is another important growth area, a set of traits that we need to focus on in the future. Antioxidants would be another good example. Traits that we don't yet use in breeding, but we could do. And of course, it's important to remember that none of these traits, very few of these traits would be relevant in all environments. There's always going to be a G/E component. And even for something, let's say heat, there are many different types of heat. Early heat, late heat, heat shocks, warm night temperature. Here's an interesting observation. We all have a good idea of what is a optimal day temperature for different crops, but we don't know what is an optimal night temperature. Nobody has even looked at that. I guess it's because we prefer to work in the day. It's sort of like that above ground, below ground thing. We know a lot about the above ground plants. And whether you're talking about physiology or pathology, we know a lot more about above ground pathology than below ground pathology, right? And I think it's the same with something like respiration. Anyway, G/E, here's a great example of G/E. Here's a set of breeding lines that were grown in the same environment with the same sowing date in Obregon, and almost the same average yield if you look at these two axes. The only difference was the way we applied the water. So one had a gravity, so essentially the equivalent of a full profile of water after a monsoon, and the other had drip irrigation, so more similar to the kind of environment you would have here with rain. And notice that the R square is 0.01. So one environment explained 1% of the other environment. Same sowing date, same everything except the way you apply the water. That's G/E. That's kind of annoying actually, but it means that you need many different breeding approaches for within a set of environments we would call drought. And then of course there's G/G. And if it weren't for G/G, the interaction of genotypes or traits, let's say, with genetic background, then probably we wouldn't be even having this conference. That's what really, or any plant breeding would be easy without this interaction with the background. And so that's what I want to talk about a little bit now. So germplasm, that's another. So obviously, which traits do we use is a function of the environment, but it's also a function of the kind of germplasm you're looking at. So I'm going to show here some work we did looking at This was with I see Suk there. So we're working with Suk, screening genetic resources as part of the Seeds of Discovery project. This is actually what I call our bottom-up approach to screening genetic resources. Not only because, well, mostly because you really do have to work from the ground up, because with that kind of number, there's not a great deal you can do. It's pretty primitive, in fact. So what kind of traits are we measuring when you're screening huge numbers? A lot of visual stuff. Maybe yield if you really want to work hard at it. But you can I'll just show you a picture of what that looked like from the air. So we did use a drone, but pretty much just to take a photo. You could probably analyze some of that data, but you can do it quite easily with your eyes as well. But the point of this kind of screening is to end up with panels of diverse but promising lines which you can use for research and for pre-breeding. So here's a couple of sets of germplasm that we've derived from that. Well, one of them is the primary synthetic panel, which is derived from a total of 2,000 primary synthetics that we developed at CIMMIT. I'm sure I don't need to explain what a synthetic is to this group. Good. And here's an example of the kind of traits we measure in synthetics, because you don't really want to drill down too much in synthetics. Synthetics are the woolly mammoths of the wheat world. If you go in and measure in too much detail, it's not that relevant, because these are non-threshable, these are tall, have strange adaptation genes. But there are some useful things to measure. And one of them is biomass. You can measure yield. And here's an association under fully irrigated conditions of yield versus biomass. And you don't see a particularly good correlation here. And often you don't in favorable conditions. But what you do see that's very interesting, these are synthetic derived checks, Vorobay and Sokol. And they have higher biomass than the elite material like Borlaug and Weeble and so on. But what's very interesting is that some of these primary synthetics have a lot more biomass. That's a trait that in a breeding program is not typically measured. In physiology, it's a trait that we would consider a very basic trait. And the point is that if you're getting 3 or 4 tons more biomass in a primary synthetic, that's all indicative of radiation use efficiency and indicative of yield potential. These are traits. This is a trait that's telling you it's a very integrative trait, but you've got something there that with the right breeding, with the right photoperiod and fertilization and earliness per se genes, you can get into yield. And you see that not just under yield potential, you see that under heat stress. These are the checks. There's all the extra biomass. Now you're starting to see a correlation with yield, which is more typical under stress. The relationship between yield and biomass is stronger. And here's under drought. It doesn't matter which environment. This is a subset of 160 lines. You're seeing material there that has more biomass. So in a very exotic set of material, the kind of traits you look at can be different to the traits you'd look at in an elite measure. You're looking at more basic traits that have promise. And here's the high biomass association panel. This is a panel put together by Hemmer to look at radiation use efficiency, to understand that trait better. And again, we're looking at let me change tack here. So the high biomass panel is sort of the introduction to the next point, which is that the trait you look at, obviously, as well as environment, as well as the germplasm, the traits you look at are going to depend on your breeding strategy. So the last point really on which traits. So here's where I want to introduce this pyramid structure. So now what's this all about? Well, here's a pyramid. At the top of the pyramid is yield. And this is the trait, the favorite trait of most breeders, for very good reason. It's the one complex trait that a breeder will always measure. And one of the reasons that you can measure a complex trait is because you have a machine to measure it called a combine harvest, right? A lot of complex traits you wouldn't measure because we don't have machines, high throughput machines to measure them. But yield is a function of biomass and harvest index at the highest level. And biomass is a function of light interception and radiation use efficiency. And then below these, a whole lot of other traits: Robisco, the Calvin cycle, vigor, stay green, canopy architecture, spike photosynthesis. You can go all the way down to the genes. And on the other side, harvest index is also a function of grain set and grain size and spike fertility and grain weight potential and sucrose and water-soluble carbohydrate remobilization and tiller dynamics and starch synthase and phenology and all the way down to the genes. And so the point of this pyramid model is that one way to make a breeding strategy is to start looking to break down yield and to start making strategic crosses so that you are deterministically accumulating traits that theoretically should improve yield potential. And in the pre-breeding program of physiology, we focused a lot on this idea. What we call it is balancing the source and the sink, which is a very old idea. It's not a new idea. It's been in the literature for decades, this idea of getting the right balance between source and sink. And it's not a particularly hard concept, really. It's the same as the economic concept of supply and demand. To optimize the economy, you need to balance those two factors out. If one becomes limiting, then it affects the other one. And so we made a lot of strategic crosses where we're looking for sources of good biomass and crossing, for example, to sources of good harvest index. So this is another way to determine which traits you should measure. If you have a breeding strategy, this is just one strategy. We've been using it quite a lot because, as I say, typically in a conventional breeding program, these traits are very often not even looked at, even the first layer below yield. And the reason is because biomass, while we do have a machine to measure yield, we don't have a machine to measure biomass. In our program, we still measure mostly the biomass by hand, which means, because it's late in the season and the straw is brittle and the spikes will shatter, it means getting up very early in the morning, too early, and out there at the crack of dawn, harvesting, so that you can estimate biomass in a way that, without shattering, and so on. And it's labor intensive. And so, again, you need to have a strategy if you're going to measure these kinds of traits. Maybe we can estimate these characteristics with drones and LiDAR and so on, but we're not there yet. Certainly not with the precision. So with that in mind, with the idea of crossing sources of high biomass and crossing with sources with better harvest index, or any of the traits that contribute to those more integrative traits below that in the pyramid, we screen and we see large variation for biomass. So that's where the high back panel comes in, the high biomass association panel that I showed at the end of the diversity set. And here you can see different sets of germplasm that we're looking for. For example, here is the synthetic panel, primary synthetic panel, a large range. Notice that the high back panel, as was intended, has the highest mean value for biomass at maturity, and there are other panels showing less and less. And this is the biomass range that we see compared to, I believe that is Borlaug, which is our standard high-yielding check. And we see biomass considerably higher than that value. So this is yield potential. The high biomass panel, it says here, were 69% of the line showed higher biomass than the best check. Some real potential there. So looking at the other side of that pyramid, harvest index, again, same panel, big range in harvest index, going from 0.4 to 0.5. If you think about it, if you just increase harvest index from 0.4 to 0.5, that's a typical range of harvest index in elite material, in fact. But you increase yield by a massive 25%, just going from 0.4 to 0.5. That's straight arithmetic, right? I mean, 25%, that's many years of breeding. So if we can crack these kinds of problems, then we potentially can raise yield. It's clear when you look at the biomass that the yield potential is there to go up at least 20% or more. And then there are other traits that are interesting. You know, this gets more into detailed physiology. A trait like fruiting efficiency, which is the number of grains per spike dry weight, or spike partitioning index, which is the investment of biomass in the spike at flowering compared with the rest of the structure, not including the roots, of course. And you see these are traits highly related to yield. And then you see some traits that are negatively related to yield, like the length of these internodes. These, as the stem is elongating, it's elongating. Stem internodes 2 and 3 are elongating exactly the same time as the spike is growing. That's a competition. And that's why you see a negative relationship, a strong negative relationship between the spike, or rather yield, and these are the spike-related characteristics, and the length of these internodes. So one of our objectives is to look for sources that have good yield, but also a low investment in these internodes, so that there will be less competition with the developing spike. And then, of course, I'm not going to go through this whole. Is there a clock anyway? I forgot my watch. Can somebody lend me their watch?
9:20.
Oh, okay. I've got till 10. Is that fine? I think so. Okay, doing okay then. So, I mean, which traits should we measure? I'm just giving you a few. I would say lodging. Lodging is a good one too. It's estimated that 10%, 5 to 10% of all the yield in the world of wheat is lost every year to lodging. But we don't really work on lodging, which is a pity. Not many people work on lodging. And yet, well, as I said, Francisco and our team, Pancho, he worked to develop this model for lodging-resistant plants. It depends a bit on probability. It's an interesting one because he worked with a group. He did his PhD in Nottingham. He worked with a group in Birmingham, structural engineers. And they said, yeah, we'll help you build a lodging-proof model. Based on, so what they work on is, for example, earthquake-proof buildings. And it's the same principle, if you think about it, because a building is tall, and if there's, even the wind can cause a tall building to move, so it's the same as a spike, same as a stem of wheat. But actually the anchorage is the same as building, the same principles are applied in this model to, as when you build the foundation of an earthquake-proof building, because the ground can shift. So these principles were put into this model. That's cross-disciplinary, right? We think we're doing great if a physiologist talks to a breeder, but actually here's some guys, biologists talking to mechanical engineers. That's really nice interdisciplinary work. And so they developed some simple tools to measure the stem strength, and you can just simply measure the root plate spread. That gives a pretty good indication of how stable the plant will be in the ground. And then you can look at the genetic variation for some of these traits and the target idiotype here and the best observed value. And you see we fall short on stem strength, we fall short on root plate spread in particular. So I bet Lee has some ideas there. And height, we're doing okay for height, I guess, but there's still a big range. And these graphs, what these graphs are telling you, okay, so in the International Wheat Yield Platform, we're talking about raising radiation use efficiency putting more biomass, putting more of that biomass in the spike. But of course that changes the moment. That makes a plant more lodging susceptible because you're putting more weight at the top of the plant, the moment, etc. And so what these calculations are looking at is how much of that extra radiation use efficiency that you would like to put into yield, you actually have to put in the roots and in the stem in order for that plant not to fall over. So it's a little bit too simple-minded to say, yeah, we can raise radiation use efficiency X% and get that much more yield, because actually, no, you're going to have to put some of that into the plant to ensure that it has structural stability. So, when you start digging down, it's not as straightforward as we would like it to be. But then nothing ever is, right? Okay, so that's a little bit about which traits should we measure in the field. I think most of those traits that I talked about can be measured in the field. We'd rather measure it in the field because that's where it has some meaning. But of course, not always can it be done in the field. Rubisco specificity has to be measured in the lab, for example. So hopefully it ends up being measured in the field. So now let's talk about high throughput and precision phenotyping, because this is interesting in the sense that high throughput is definitely the trend, but we mustn't lose sight of the fact that we're not going to be able to measure everything at high throughput, even if we want to do that because it's breeding and it's genetic resource screening. Sometimes you have to basically make subsets. That's what I was trying to imply earlier. You start off with high throughput, but you may need to end up with detailed physiological phenotyping on a promising subset of material, just because you can't measure it on everything. But even before we start with that, we also got to remember that we have to have the right kind of population. Here is a mapping population. These are two sisters. These are sister lines. This is the problem with wheat being a hexaploid, right? These are two sister lines from a mapping population from Australia, where and this is random. This wasn't the earliest and this wasn't the latest. These two happen to be sitting together by the randomization. And this line is clearly approaching physiological maturity, and this one is approaching heading. And tell me, how do you phenotype that? How do you get any meaningful phenotypic data from that kind of population structure? I make the analogy that it's like giving the same IQ test to a 5 and a 50-year-old person. You don't. And so you can't really phenotype that. And that was the problem we were struggling with 15, 20 years ago, especially with respect to mapping traits, that it's easy to make a wide cross and say, oh, there's tons of polymorphism. Yeah, damn right. But it's uninterpretable, because all you come up with here is PPD and VRN, and we showed that in studies with Peter Langridge. So we developed a population called Ceribabax. It was designed to not have that large degree of confounding effect from major genes. You're trying to detect, in this case, well, it doesn't matter whether you're trying to detect QTLs or just trying to get some meaningful information to make a cross. If you have this kind of confounding effect of phenology, it's very hard for any of the information to be useful. So you need to narrow it down, and we developed populations like Ceribabax and the WAMI panel, which is Weird Association Mapping Initiative. I'll show some data. And basically the idea is to control the genes of major effect. So height and phenology, because usually when we talk physiology, we're talking about complex traits. And I think we have a good enough understanding of some of the genetically simply inherited traits that we don't need to be so sophisticated in our measurement. We can actually, when I joined CIMID, Tony Fisher, who many of you know, he was my boss and director of the wheat program, and so this was ages ago, and carbon isotope discrimination was the trend at that point. We're flying down to Mexico together by chance to start my postdoc. I was like, can we buy a mass spectrometer? Because I think it would be really exciting to use carbon isotope discrimination for drought work. And he's like, what you need, Matthew, is not a mass spectrometer. You need a ruler and a balance. and nearly 30 years later, I totally agree with him. I didn't agree with him at the time, but you know, I didn't control the budget. But yeah, I mean, sometimes you've got to keep things simple. Anyway, so here's some of the high throughput selection tools. And the best one of all is the low resolution stereoscopic spectral radiometer connected to a supercomputer. What is that? What is the low resolution stereoscopic spectral radiometer connected to a supercomputer? What else do we know that as? Your eyes and your brain, right? That's the best high throughput selection tool. I'm a physiologist and I'm saying that because it's true. That is, but we don't see everything. We don't see temperature, and temperature is a very useful trait to see. And we don't have the resolution that a spectral radiometer has. So your eyes are low resolution. In other words, you can see green, but can anyone here see how much carotenoid is in the leaf against a sea of green? No. The only time you see that is, for example, in the autumn when the leaves lose their chlorophyll and they turn beautiful red, orange colors. That's the carotenoids. And they're important pigments. And spectral radiometer will see every single nanometer and it'll give you a number, quantitative amount. So, you know, we don't see certain parts of the wavelength. I mean, light is broad, right? And we don't have the resolution. And obviously, high throughput could be markers. So that's an important point to remember. And then at the other end of the spectrum, and these are high throughput things, we have very low throughput things. So this is related, I'm not going to explain this whole slide, but this is methodologies to estimate air photosynthesis. Here, this is an infrared gas analyzer. This is a special sleeve that was developed, and this is a cuvette that was developed to strap around the spike and then cover it with LEDs of photosynthetically active radiation. That's low throughput, still in the field, but low throughput. And then the other way, you can shade the spikes, or you can spray DCMU, which inhibits electron transfer, or you can measure carbon isotope discrimination from different tissues and use that in an algorithm to estimate contribution of spike photosynthesis. So on the one hand, we have super high throughput tools like infrared thermometer, and the other hand, we are actually using in the field some pretty slow techniques. So there's a range of things. But let's go back to the high throughput. So we all know that canopy temperature is easy to use. These days, one of these instruments costs you a couple of $100. It has an integrating function, so you can move it over the plot. You're not measuring one leaf at a time. That was the problem back in the day when physiologists like Tony Fisher started working. They would show up in the field with an infrared gas analyzer the size of a small truck. They would spend a week calibrating it, and then they'd spend a day getting one photosynthetic reading from one leaf, and then turn around to the breeders. I think Tony Fisher said this to Borlaug, just after he'd won a Nobel Prize. Yeah, we don't really need breeders anymore. Well, maybe he didn't exactly say that, but they definitely had some disagreements. Things have moved on, and now you have instruments like that, where you're not measuring one leaf. And you don't need a week to calibrate it, and it's tiny and it's inexpensive. And you look at that beautiful relationship between canopy temperature measured under warmed conditions and yield, explaining half of the variation in yield. And that's a physiological trait. There's no autocorrelation with grain number or grain size or anything. That's pure physiological trait. That's a function, and most certainly, of the vascular system, the matching evaporative demand, due to the vascular system, and probably some source zinc balance thrown in there. Measuring the temperature of a plant is a sort of the equivalent of a doctor measuring your blood pressure. That's the first thing a doctor always does, right? They measure your blood pressure. It seems a bit odd, really. But they can tell if your blood pressure is too high, they know a set of things are wrong with you. If it's too low, they know there's another set of things wrong with you. If it's perfect, they know that you probably shouldn't even be there, because you're probably just faking it and trying to get a day off work. So temperature, that's a very, very useful tool. And I'll tell you another reason why it's useful. Not only does it correlate with yield under heat and drought stress, it also correlates with root depth. So this graph is showing us that yield under drought, this is work from Marta Lopez when she was with us in Mexico, that the more biomass in the deep part of the soil profile between 60 and 120 centimeters, the higher the yield. And the more root biomass in the deep part of the profile, the cooler the canopy. So in other words, and look at that R squared, 0.88. So in other words, a horribly laborious trait like root depth that would normally, you would need a piece of hardware like this and a lot of time and a lot of root washing to measure. I should have a photo of people washing roots, actually. That's no fun. Anyone who's ever done that knows. Can be measured very simply with an infrared thermometer. That is a very important innovation. I still don't understand why there's such a huge investment in tomography, people working in lysimeters to measure roots, and yet we can estimate roots so simply with an infrared thermometer. And that was then, this kind of approach was used to find genetic basis. So that's an important aspect of phenotyping. And obviously, this is where that trade-off between high throughput and precision and low throughput comes in, because typically when we try to identify QTLs, you want, or genetic basis, you want lots of lines, so high throughput is better. So canopy temperature helped us here. You're trying to get a genetic basis for, yeah?
Like you mentioned that you are measuring yield photosynthesis.
I'm going to come to that in a minute, yes.
Yes, to see how much does it go from yield photosynthesis, but then also probably you are measuring plastic photosynthesis. Because according to some, there is 70-80% shows on leaf, flat leaf, and 20-30% on field photosynthesis.
Yeah, I mean, that's one estimate. There are different estimates. Yeah. I'll come to that in a minute. This is so using canopy temperature to screen the Ceribabax population, we were able to identify, this is work from one of our PhD students a few years ago now, even common QTL across heat and drought. So heat, hot irrigated, so loads of water, drought, receding moisture profile. So a nice finding. And this turned out to be related under drought. So as we saw in the other graph, more investment of roots deeper in the profile, below 60 centimeters. But under heat, not quite the same, just a bigger investment in roots altogether. So that sort of makes sense. Under drought, you want to find water down in the subsoil, because that's where the water is. But what about under hot irrigated conditions? We weren't really sure what to expect here, so that made sense. Because it's a bit like the analogy of staying in a cheap hotel and getting up after everyone else, standing under the shower, turning the tap on, and no water comes out because everyone else is taking a shower. That's a not very good vascular system. So that's a plant with warm canopy temperature, a very high demand, which is everyone taking a shower once, or in the case of a plant, very sunny conditions, dry air. If you don't have the roots, enough roots to match that demand, as in this case, then you're going to have a warmer canopy temperature. So think of you can think of it in those terms. Okay, so spike photosynthesis. Why is spike photosynthesis interesting? And why would we even think about it? Well, that photo can sort of tell you a lot right there. Which organ is the first one intercepting the light here? And how much of the light is this organ intercepting? The spikes are the first organ, and they can be intercepting anything up to 40% of all radiation. And we measure leaf photosynthesis all the time, but we don't measure spike photosynthesis. There's very few papers. There's thousands on leaf photosynthesis and a dozen or so on spike photosynthesis. So that's out of balance. But there are reasons, and always we shy away from difficult things, which is why Lee asked me to talk about this, presumably. The spike, so a leaf, we think of a leaf as a two-dimensional object. But the spike is a three-dimensional, there's no way around the fact that the spike is a three-dimensional object. And it has a very complex geometry. That's why we typically find it difficult to standardize the units of photosynthesis. That's the easy part. The hard part is that the way we measure photosynthesis is with gas exchange. So you're measuring the fixation of carbon dioxide into Kelvin cycle. So you measure that flux with a gas analyzer. But in the case of spikes, they're very sneaky. They're actually not getting that gas from the atmosphere. They're getting it from respiration internally. So you can't even measure that with a gas analyzer. Not directly, not in light. You have to measure the respiration rate in order to be able to estimate how much CO2 they may be fixing. And then they may be fixing some more. So it's all pretty complicated. And this is why, going back to this, Emma's technician built something to illuminate the spike all the way around, so you don't have this one-sided effect. And then, as an alternative, keep my eye on the time here, as an alternative, this will only ever give you a snapshot. It's like taking a photo on holiday, everyone is smiling, right, in the photo. But does that mean everyone was smiling all the way through their holiday? No, they might have been fighting, they might have been crying, but you don't take a photo of that, right? You just take this snapshot. And when you measure gas exchange, you're measuring it on 10 minutes from one day out of 30 or 40 days of grain filling. But so we came up with another way to have in a more integrative way to estimate spike photosynthesis, which is to wrap the spike in a textile, so the gas, the ethylene is not accumulating and so on. Still gas exchange, but no light is getting through. And that way you can get an integrated estimate of the contribution of the spike by measuring the difference between a check and this spike that was covered with something that stopped the spike photosynthesis. And we used that to do a QTL analysis and came up with some QTLs for spike photosynthesis. As far as I know, the first ones. So I think I'll skip over that one because it's probably a bit behind. So I think I wanted to give you a taste with that last section, 10, okay, of high throughput versus precision phenotyping. There are definitely very useful things we can measure that are high throughput, and I'll come a bit more to that in the next section, like canopy temperature. But there are things that if we think are important, like spike photosynthesis, and there's every reason to believe they are important, we don't know what is the genetic variation for spike photosynthesis, but we do know that the spikes intercept a lot of radiation, and they have some important heat and drought-tolerant characteristics. So sometimes we have to bite the bullet and say, okay, it's not going to all be high throughput and easy. Sometimes we have to go the hard way. Okay, choices among technologies, handheld UAVs, phenocards. So briefly, we saw this. It's some easy things to measure. And most of what I described so far in terms of high throughput is related to these ground-based, everything I've described so far I think is related to ground-based measurements. And, it's been great. Here's a study from Jessica Rikowski, which showed how when you combine genomic with phenomic data, you predict, in a genomic selection type model, you're getting much better predictions of yield. You know, a very nice example of combining the genetics and the phenomics. And that's all ground-based. But we can go to the air, and we've gone to the air, and here's Francisco, and here's him having fun with his drones, and here's some data from his predecessor that showed that when you compared ground-based and drone-based data, you know, as you'd be kind of worried if you didn't see an association, frankly. But you're also basically getting better data from the airborne platforms, or explaining more of the yield or the variables that you're trying to explain. Here's a graph showing how radiation interception can be estimated with aerial NDVI. Estimating radiation on the ground is slow. You need a light bar. It's actually hard on your back because you're bending down all the time. It's slow. And explaining 60-70% of variation in light interception. Whether that's the important part or not remains to be seen, but making progress in bridging that gap between what previously were hard things to measure, slow, using aerial approaches, 3D construction of plant height and structural parameters. Ideally, we'd like to estimate the volume of plots, so that might be a way to estimate biomass, a combination of volume and yield and height. Well, height would help you with volume. You know, maybe we'll be able to explain 80% of final biomass. I'd be happy, because I wouldn't, and my team wouldn't have to get up so early in the morning. And one of the reasons that you get better data from or more precise data from the aerial platform is because with an image, you're able to go into this image and correct using pixels. You make a frequency distribution, you say, well, these are the pixels that are the most representative, and these extreme ones, you know, a dog got into the plot, or there was poor establishment, or who knows what heterogeneity of the soil. So you can clean up, as well as the fact, obviously, that when you're walking, it takes time to get from one end of the field to the other. Sun angle changes, temperature changes, so that introduces error as well. So these are some of the reasons why you would expect the high throughput, the imaging approach to explain better your variables. And then, so now we're talking about these options. So the aerial approach has a lot of advantages, but it has, as I mentioned at the beginning, a lot of disadvantages too. And this is the kind of image that you get when you take a photo from the air. It's a bit scary, isn't it? And you need to be able to use these techniques of noise reduction and calibration and so on to correct that image, because that plane is moving, right, to this image, which looks more like what you would expect to see. And that, I can assure you, is not something that any of us can do without a lot of training. And this is the drawback, and what worries me a little bit about the love affair with drones, because drones look like fun, but most of us would not consider it that much fun to have to correct these kinds of images. It's just, you know, maybe for some people it would be, but its point is it's a specialized task. You need a person who can do that. You have to probably hire somebody with those skills already, pay them a lot of money, etc. So the high throughput, in theory it's more efficient, but actually in practice it may not be. Unless you're really ready to embrace and have the budget to do the whole work that's involved with image analysis and correction of those images. And so in the middle you have these sort of phenocarts and gantries, somewhere in the middle, where you may be using the same kind of sensors as we used before. This is Jared, who worked with us in his PhD, and he just strapped these instruments that we would typically walk around in our hand to one very simple, it looks like a circus unicycle, but it's actually a bicycle sawn in half, so it doesn't have to be high budget. And he's just strapped these instruments here. So basically, I mean, if we had 4 or 5 arms, it's the same thing. And so I like this as somewhere in between the traditional handheld and the sophisticated gantries and drones. Drones are great if you're in an irrigated environment because you can even measure when everything's wet and you can't get in. But you know, now in Mexico we're getting a lot of grief from the authorities. Have you registered your drones? Do you have a pilot's license? And I think all of us are struggling with these issues too. So, you know, I think it's worth considering these intermediate steps too. And don't forget, here's a great figure showing the strong relationship between yield, farmer's yield in the Yaqui Valley and night temperature. Don't forget, when we get sophisticated, let's not forget the basics. Because CIMMYT, like many research programs, we don't prioritize getting simple things like metadata. It's crazy. But everyone I speak to, they're like, oh yeah, metadata, yeah. Yeah, we're not actually sure who's responsible for getting metadata. It's crazy. But metadata is so powerful. R² of 0.75 between yield and night temperature. Crazy. And yet we don't. Why don't we prioritize it? Because probably a donor, if you said to a donor, oh yeah, we want to invest $50,000 in measuring metadata, they'd be like, oh, how boring. Metadata. That's, you know, there's nothing novel about metadata. Okay. I'll get off my soapbox. Okay, so lastly, case study very quickly. So we do all of this in order to provide useful information that breeding programs can incorporate, but at the same time we need to prove that this is valuable. And because breeders are not always willing to test your hypotheses for you because they're like, we're busy and we've got to generate 1,000 new genotypes this year and last year and next year, so whatever. So what are the objectives of physiological pre-breeding? Obtain, first and perhaps most important, obtain proofs of concept about which physiological traits or allele combinations can boost yield in a realistic breeding context. Also broaden the genetic diversity of the gene pool through selective use of genetic resources, and I emphasize selective because you can't just use anything, it has to be stuff that's already phenotyped. And then of course deliver novel sources of traits and yield potential to breeders worldwide in acceptable agronomic backgrounds. So that's where we're going with this. Here's conceptual model for the strategic crosses for yield potential. We use all of these traits. Don't have time to go into detail, but we can come back to it if you want. Similarly for heat, we already looked at that one. But notice on the previous one, these traits are categorized as source or sink, pre-grain filling, source and sink during grain filling. And remember I said that is our strategy, to combine a good source, so think biomass, with a good sink, think harvest index, and all of those component traits that are related to that. And similar for heat, here's again the model. And to cut through, so we found sources of these traits using all of the techniques that I've described, and then we made pre-breeding pipeline, which is different to what the breeders do because we're trying to get a very quick result. We're not trying to produce a finished material with quality and full spectrum of disease resistance. We essentially want a quick and dirty outcome in order to test specific hypotheses. And so these are the traits these are the sites where we measured these traits. And yes, as you would hope, otherwise we're really wasting our time, we're getting some lines. This is an average of 32 sites. We're getting some lines that, you know, marginally better than the best check. But what's really interesting about this result is two things. First of all, although the yield isn't that much higher, the biomass is much higher. And this is exactly what you would want out of a pre-breeding exercise, because putting that material with a pretty decent yield, you know, competitive with the checks, but 2 or 2.5 tons more biomass, that can be very useful to a local breeder, because if they get that adapted to their environment, that 2 tons or half of it can potentially go straight to yield. And the other thing that's interesting, if you look at the pedigrees of these lines that were, marginally, or let's say statistically similar to the Czech. Look at the pedigrees, Mexican landrace there. Now, these in the green all have both a landrace and primary synthetic in their background. And some just have a synthetic, some have a landrace. But the point is, every one of those has some new genetic diversity that was deliberately introduced for expression of complex traits. So these are two achievements, and as I say, based on a cluster of 32 sites, fairly convincing outcome. And that was recently published in Euphidica. Some of you were co-authors on that study. And I think I'll end there. So a lot of our protocols are published in these manuals. They're downloadable, or you can write to us and get them. So if there's any questions, I'd be happy to take them.